So I don't have any self-introductory slides. So I'm an assistant professor in the University of British Columbia and a senior research scientist in Vancouver Prostate Center. Most of my research has been on sequence analysis. I've worked extensively on sequence mapping and sequence compression and structural variation from small and large. So today I'll be talking about the detection of the genetic variants in cancer genomes. And I'll have like 3 parts. I'll just go briefly over some background, a little bit of a biology. I'm not a biologist, so I won't be going too much into detail. And then I'll talk about single nucleotide variants very briefly, and then to the structural variations. So basically, genomic variants can be categorized into two classes. Single nucleotide variants, where your donor genome has a single change against your reference. And we have structural variation, which are the larger changes. And you can see like there are different types of them: deletions, insertion, duplications, inversion, and translocations. Why are these so important? These aberrations in basically coding regions can generate novel proteins. And these proteins can have a high impact in the phenotype and more likely to cause diseases. Many of these structural variants are basically associated with diseases, especially in cancer. In context of a prostate cancer, there's a huge literature. For example, Tempers ERK in prostate cancer, which I've observed in like 50% of the patients, is an upregulating of the oncogene. And those fusions, the patient which has those fusions has a higher morbidity rate, and so on and so forth. So a little bit, I just grabbed this slide from a paper, and I mean this is one of the paradigms of precision oncology, but there are many of these pictures you can see. But basically we start with a biopsy, and when we do sequencing, genomics, transcriptomics, we do some analysis, we do functional testing on the xenografts, etc. We do treatment with the drugs, and then again, as continuation of this process, we have to recontinue biopsying and doing this process and modifying the treatment if needed. So challenges if you have to do this with tissue biopsy. The tissue biopsy in general is an invasive procedure. It's very painful, expensive. You cannot really repeat it many times. And when we're dealing with metastases, usually it's a multifocal. That means you have to biopsy multiple lesions in the body, which is very invasive. And tumors in this situation of the stress and in response to treatment, they evolve rapidly, which makes the repeat biopsies desirable but impractical. So liquid biopsy, which is a sample obtained from blood or urine, recently has shown the great potential to be used as non-invasive alternative for genomic variation detection and disease progression monitoring in cancer patients. So this is not the first time the liquid biopsy being used. Probably you have heard of the non-invasive prenatal test, which is basically using the same idea, using the cell-free DNA to find the trisomy diseases in the fetus. So what is cell-free DNA? Cell-free DNA is DNA that's naturally occurring in the blood. It is released from the cells that are going under apoptosis or necrosis. And basically, a portion of this DNA that is released into blood is coming from the tumor cells. We call them circulating tumor DNA. And how the sequencing has been done, one of the important things about this cell-free DNA, they have a very short lifespan in the blood, and usually they are protected around nucleus and position. So basically, usually what's been done in practice is the target panels. You just consider some genes that you're interested in. You just extract the DNA, this cell-free DNA, and then you do PCR because the material you're extracting is pretty low. And then you do capture. You, for example, are interested in gene A, you just capture that one, which goes through another PCR process to increase the material for sequencing. At this point, you have a sequencing data that you can work with. What are the challenges of working with such a data? Well, one of the important one is the low dilution. So the tumor content in cell-free DNA can be pretty low. It has been reported to be like 0.01% of the whole the content is just a tumor. Heterogeneity in cancer is another big problem, because now consider the low dilution. Now you have subpopulations there that you're going to have smaller percentiles. Probe efficiency in the capture, when you're doing the capture, these probes are not as equally efficient. So you may end up with non-uniform distribution of the coverage. Obviously, sequencing error is always there. And as you see, we had like number of the PCR rounds, which is, in our experience, we have done like 14 rounds of PCR pre and post capture, which automatically introduces more error to the sequencing data. And the other thing is, these data, we never shear them. We never fragment them anymore because they're very short. Usually they are less than 200 bases. Like we have median of 167 and shorter as well. So the first is how to detect nucleotide variants. This is mainly work of John Kochkan, who was a master's student at the time. And now he's a PhD student. I'm not going to go too much into detail because there's a huge work, and most of you guys are very familiar with the single nucleotide variant detection. So most of the single nucleotide variants basically start with some pre-processing and some post-processing, and this is what matters mainly in calling high confidence variants. So to just give you a little bit of a detail, basically you just pre-process reads, you just remove the trims, trim the adapters, map the reads with any popular mapper you can use. You do recalibration and error correction. I'll talk a little bit about this one because these are short fragments. Whatever technology we use these days, there are longer reads, like 75 bases, 100 bases. You will end up with overlapping reads. So you can actually maximize the information from these overlapping reads to try to recalibrate the quality of the reads in those regions. And obviously, you calculate the basis statistics on the genome. For this one, we use a very simple model. We modeled the mutation as a Poisson distribution, where K is the number of the reads experiencing the mutation, and R is the average error rate for each position which is coming from the platform, and the N is total number of the reads covering that region. And based on that, we obtain a probability that this mutation is being real and not an error, and we provide a confidence error, which is basically Phred score-like type error Phred type score. So finally, we have, as many other tools, we have some post-processing. We just eliminate those mutations which we don't believe in them, which has probably low number of the read counts. They have strand bias, which is very, very frequent. That if there's imbalance in the number of the reads coming from different strands, we can eliminate them. Homopolymer region filter, which is basically another error which is very common in the Illumina sequencing. And finally, this is the most important one, is the singleton noise ratio filter that I'll show an example. This is an example of 5 prostate patients and 5 loci, and all has been called as a mutation. But reality of the fact, the first 2 are really mutation, because you can see in other patients there's nothing. They are really standing out. Their allele frequency is about 8 to 10%. The next 2, all patients experiencing the same mutation. If we call this as a mutation, this is more of a sequencing error, or like a PCR error than mutation, because it's happening in all of them in very, very low allele frequency. And this is one of the filters that's take care of like number of the samples together. So just trying to remove the batch effect. And the last one is just all of them has the mutation, but they're high allele frequency. They're like a recurrent mutation that's happening in the patient. So as David and I think Sergey talked about simulation, we have done simulation. Yes, we beat everybody else. But we didn't stop there. We tried to just do multiple real samples, and we tried multiple sequencing platforms. This is just, I'm going to show one example and not going to talk about the results. But this is basically a mixture of the 2 different cell lines. We mixed them based on that proportion 10 to 1, 20 to 1 to 50 to 1, which is basically 10%, 5%, and 2% mixtures. And they were like validated mutations. This is one of the best, actually, at that point, working on the cell-free DNA we tested. The other ones, they were not optimized yet for this purpose. And you can see which ones are calling the mutations. As your material decreases, the noise goes higher. So we still start losing the mutations as well. So the question is, can we do better? Can we improve on such a low allele frequencies? And this is actually work of a master's student of my Bara. which presented this work in Recombsing last year. In fact, there can. So we have something called unique molecule identifier. These are basically small, very short DNA fragments that we can ligate to the molecules, actual molecules. And then we can just go through the same process of PCR, capture, and PCR. So how can this help us? So this is the final product, the PCR product. If you go through the sequencing, what will happen? You'll observe that there are sequencing errors here. But if we hypothetically manage to cluster these, if we know that they're coming from the same physical molecule, we can easily just fix this just by just looking at the same location. But it is not very trivial because the UMI itself, this barcodes that you're attaching, can also undergo this mutation through the PCR cycles as well as the sequencing. So how are we going to solve this problem? We try to build an unweighted, undirected graph of whole reads. Basically, each node is a set of reads with identical features. And I'll get to the features, what the features are. And an edge exists between the two nodes if they have similar features. So the feature for a read is basically two segments. Number one is a barcode. So these DNA fragments that we attach, we just grab them, concatenate, and we obtain a barcode. That's called a barcode of this molecule. Okay, that's one of the features. The second feature is based on, we're going to call minimizer. So minimizer, if you consider, given a string and a k, a minimizer is the smallest lexicographical k-mer of that string. Okay? And this hasn't been used in other contexts like compression because they're fast. So for example, if you consider this sequence and you have a k of 2, TC will be its minimizer. So we'll extract number of the minimizer from the read. We just subsection the read into multiple segments, and from each segment we pick a minimizer. Now, how are we going to just build this graph is basically we are going to use locally sensitive hashing to find basically these nodes with similar barcodes. Because barcodes can undergo sequencing errors, we consider like 1 or 2. If it is more than that, this one will be easy to do because it's just going to increase your running time complexity. So probably all-to-all comparison would be better. For the single error, we just use like number of templates because you have 4 bases. You can have 4 different locations that can have an error. So here, this is the first template. Basically, these all considered for hashing. So basically here, the 2 reads have a similar barcode, ATG. But if you look at their minimizers, they are not. So they don't have a similar content in the sequence. So they cannot be actually connected together. So we can repeat this one. Actually, here, there's one example that's going to be connected. So here, this is the second template. These 3 have same minimizer. But if you look at their k-mer content, both of them have the same k-mer content. So their sequence similarity is very high. So basically, we'll add basically a read, an edge between the node 1 and 2. And so on and so forth, you can build this graph. So what we're going to do next is basically, from this graph, we'll find the connected components. Because these are basically the set of reads have very similar content and barcodes. And based on this, we'll just do a multiple sequence alignment. For this, we're basically using a partial order alignment, which is pretty fast. It's a heuristic. It's pretty fast. And then we build the profile, and we can actually fix the sequencing error and the late cycle PCR errors. So does it have any effect on the downstream analysis? So simulation has been done. I mean, there's extensive simulation, and there's a source code you can test it yourself. But we tried on the real data set. We grabbed a genomic standard, HT701, which has number of the verified and reported mutations. We target a 36 gene panel covering about 240K genomic regions. And in this panel, there were 254 reported SNVs in the literature, and 10 of them is actually validated through using the ddPCR. So we used 16 base barcodes, 8 plus 8 on each side, and generated 22 million reads with Illumina. So we modified a number of the clustering methods out there for this purpose to be able to compare what will happen. So if you remember our model with the Simbic, we actually managed to grab the error model of the platform. Error correction on the reads actually improves the error model of the platform. So you actually can say error model is lower, so you can actually go for more sensitive calling. So this is original one. If you just normally call the reads without considering any barcodes, you obtain about 2,000 mutation calls. From there, about 227 or 8, I can read it actually, is in the reported list. Now, if you use any of this clustering method and reapply it, the number of the mutations drops significantly. That means it's actually compressing this information into physical molecules to improve heavily. And then, obviously, here, the number of the actual validated or reported mutation falls down. But what would be the reason for this one? We said that there's a minimum depth filter, but when you collapse them, also your number of the reads decreases, like by tenfold probably. So if you increase the, basically decrease the error of the sequencing, you'll about, the sensitivity goes high, so you report about 84,000 locations as a mutation, which is not correct. But if you look here, you actually report more of the validated without losing a lot of adding too many false positives. So this helps to manage to go in lower allele frequencies to sub 1%. But it's at a cost, it comes at the cost of a little bit more sequencing cost. Yes?
Question. So you have runtime performance over here.
Correct.
I assume the reason why you're using minimizers in an LSH is to speed that up.
Correct.
So like doing just a naive edit distance.
Yes, it will be pretty expensive.
Pretty expensive.
The reason is that this is a pre-processing step. We wanted this as part of QC, and after that we can collapse them quickly so we can run it. So in clinical application, I think that the turnaround should be pretty fast, and really these are for monitoring purposes. So one of my goal is actually to be able to just utilize them later on towards a clinical application. So having a higher speed with high sensitivity and accuracy is a must.
Yeah.
Okay, so second part of the talk is detecting structural variance from such data. This is work from the two, a PhD, former PhD student and a postdoctoral fellow that I advised. So this is an overall overview of the whole method. And if you're interested in structural variation, there is actually a short course in the next week's program, which will be presented by Dr. John Alcon. He'll just go into more detail probably on these topics. So basically, as many of structural variation pipelines, they start with extracting those reads which shows any signal of a structural variation, and then we try to build a contig from those reads which is showing the signal, and then try to find their location on the genome, and then trying to basically classify them to different classes. So there are many indicators for structural variation. So one of them basically is the split read, that you have your read is just splitting and mapping into two segments. The other one is one-end anchors. In one-end anchor case, one of your mates never maps, the other mate maps. So it could be part of a novel insertion discovery, novel insertion that you don't have it in genome, so it's not getting mapped, or it could be part of high error. Soft collaborates. This is basically a heuristic applied with many mappers, trying to just end part of the reads that it's not just mapping well. Okay. So before going there, so as soon as we extract this, there's one step which is a clustering step based on the breakpoint of this split trees. So considering this splitting breakpoint, we just try to find all the reads that just accumulating there. So that's basically called a cluster. And then what we do, we try to build an assembly of that. So we developed a strand-aware overlap layout consensus assembler, which is in practice pretty fast. Obviously, there's a lot of off-the-shelf assemblers you can use, which has many more features like error correction, etc. But there requires a lot of API call. But this is a very simple assembler that's just for this purpose developed. So the input is basically a set of unmapped reads in the cluster, and the output is supposed to be a contig which maximizes the total sum of overlaps between these reads. How we solve this problem? We solve this problem as a maximum weighted path problem in a directed graph, where each vertex corresponds to a read, and there is an edge between the two nodes if they overlap, and the weight of that is the size of the overlap. So for DAG, so the acyclic graphs, there's an efficient dynamic programming. If you have an order on these edges, you can actually calculate this dynamic programming pretty fast. So this is an example. This is basically reads, and this is the overlaps. And each of the nodes correspond to one of the reads. And as you can see, this is basically the overlap and its weight. And this is a topological sorting on this graph. And basically, when we're doing this calculation, we can go from V1 to V2 or V3. But this one, V2, has a weight of 2. So we select basically V2. And then between V3 and V4, the weight of the V4 is higher. So just to grab this one, basically, this is the maximal weighted path of 10, which is giving you this contig. Obviously, this problem, if there is a cycle in this graph, this problem is an NP-hard problem, and it can be long path can be reduced to it, so you can just prove that it's an NP-hard. But what we do, we just this is seldomly happen, so it doesn't happen too often. But what we do, we just apply heuristic. We reduce those edges in the cycle, basically having like the smallest in degree node. For example, here, V4 has an in degree node of 1, so this edge has been removed. It becomes a DAG, so you can apply this. So fine, we find the contig. What we do next is basically try to find these segments of this contig that comes from different locations in the genome. Well, thanks to Ryan, which talked about the index k-mer mapping and basically k-mers, we apply a very similar strategy because it's pretty fast. For each contig, we just built all its k-mer content, and then we scanned the genome. We found those locations which had these k-mers, and then we try to stitch them together by considering small gaps, one base gap, to obtain a bigger segments, or like we call them intervals. For example, this is an interval here of size 40 or higher, and then we use a chaining optimization basically to chain them and find a path that can explain the whole thing as a structural variant on the genome. So how this basically is done again through a graph formulation. So each of these basically mappings have their own nodes, and we add a source and destination, and the weight on these edges is basically the size of this interval, target interval. And they're connected to each other if they're compatible. So for example, here, this is an example of the mapping from the contig to the genome. And this is showing an inversion. So they're like this formula that's showing the compatibility. So we calculate the start and end on the genome, and as well as on the contig. And if they're compatible, we just add these edges to them. And then we find the longest path on this stack through a greedy approach. At any step, we try to find the basically maximum weight that we have there. And since it may have multiple solutions, we can report all the co-optimal solutions together. Finally, some of these contigs are very simple to just look at them. If they have single breakpoint, you can easily annotate them. You call them insertion, deletion, etc. But sometimes we have translocation, and this is a little bit harder problem. So we made a simplification on trying to find these intervals, or basically if you sort those, basically these are the contigs, and this is the genome, and these are the mappings that we have obtained from the co-optimal mapping solution. So we sort them, so these are basically how it looks like. We basically try to simplify that there are not overlapping structural variants. So this becomes a scheduling problem. So we try to pick up those contigs which can explain the most of the structural variants. And this is basically a set that you can see from these 4. Because if you select this one, you cannot select these 2. So basically, this is a greedy algorithm. You pick the first one. If there's an overlap, you eliminate the next one, and so on and so forth. Results, as I said, thanks to David, we don't have too many simulation results, happily. What we did, we actually tried to, again, use a genomic standard. We bought one of these bottles from the companies. We just sequenced them, and we tried many tools to see if we can detect these, like this type of deletions and insertion fusions. And obviously, we can. Some tools, they cannot. And there is a good reason for that. So it doesn't mean they are bad tools. It means that they are not designed really specifically with this type of a problem. Because there's one signal that many of the structure variants use, which is a discordant mapping, which is basically the distance between the two fragments and the orientation of, which is not really applicable in this case where your fragments are pretty short. We try to optimize all the basically their parameters to get the more sensitive results, see if they can really pick up the signal. So LAMPA, one of the incorrect one is, I'm just going to just say it's incorrect because it picked a second gene which wasn't the correct partner. That's why we called one side is correct, the second part wasn't correct. But one interesting thing is observation is actually the number of the calls they're generating. So there are too many calls. So still there are false positive available. Yes?
Quick question. So it looks like there's only 4 variants right here.
Yes. There are more variants. Unfortunately, our panel was small.
I see. Yeah, because I was wondering, what is the frequency of these sort of structural variants in, an actual patient that you see? Which is hard to say because how do you measure?
Okay, so I can talk a little bit about the prostate patients because I know a little bit more. I mean, a fusion, for example, in prostate is more common, and there are nowadays observed that there are more events happening. For example, recently there were like 2 cell papers reported a duplication of an enhancer, which is 700 kb upstream of the gene, which when this happened, you observed an increase in the expression of the AR, which is the most important gene for the prostate cancer. Also, there's one of the mechanisms that they have recently observed is actually truncation of the DNA binding domain of AR. Basically, it's a mechanism for resistance. It's also a big deletion, just cutting from the exon 4, if I'm correct, exon 4 to exon 8.
So these are like individual ones, but in total, is there like 5, 10, 100, 10,000 variants? Standard prostate cancer patient.
Well, it's hard to say. Really, I have no statistics to tell you. But size-wise, they're biggest. They're actually, there's some stats, but I cannot really recall it. Okay, I have one very interesting math problem as well. Okay, if you love LA, but you love cloud and rain, that equals to Vancouver. And as everybody, we are also hiring. So if you're just interested in coming and working in algorithms, data structure type problems, feel free to send me an email. And at the end, last but not least, there are many people involved in different phases of this project. And thanks to the funding agency for supporting this research. And I'll take questions.